Hello everyone, welcome to my biology classes. So I will be starting with phylum number 1, that is Porifera. Okay, so I have already made one introductory video from the chapter Animal Kingdom. So please go through it properly so that you get to know the characteristic features, so it is easier for you to know the phylums easily. Is it clear? So today I am going to start with Phylum Porifera, which includes all the sponges. Okay, so let's see the characteristic features of Porifera. So generally these Poriferas are marine in origin, so you can see them in marine habitat. They are asymmetrical. In the first introductory video only we have talked about what is symmetry, asymmetrical. Okay, so they are marine, they are asymmetrical, they are known as the primitive multicellular animals. All right, they are having cellular level of organization, means cells are not clubbed up together to form tissues. Okay, so they are confined to the cellular level of organization only. Okay, then food intake, gaseous exchange and excretion occurs through the water transport system, that is also known as your canal system. They are having one particular system that is called your water transport system or canal system, through which the food they used to take in food, there is some gaseous exchange going on and there is excretion through this water transport system, or you can say canal system. Is it clear? Okay, then moving on to the next slide. So you can see the diagram here. It is showing a sponge. Okay, so what it is saying, that water enters through pores called ostia. So can you see here pores? So water enters through this pores, that is called your ostia. This is your ostia. Is it clear? Then it goes out through osculum. So this here it is going out. I don't know why it is coming. Wait. Okay, it has gone. So can you see ostia? This is the ostia from where the water used to enter, through ostia. These are pores. The water used to get inside and it goes out through the osculum. So the direction is also showing it. This is osculum, where the water used to go out. Okay, via central cavity. So they are having a central cavity called spongocoel. Okay, spongocoel. Okay, this spongocoel is lined by a collar cell or choanocyte. So they are lined by a collar cell. These cells are called your collar cells or choanocytes. Is it clear? So let me clear this thing, make it more clear. Suppose this is a diagram of a sponge. Okay, rub few parts. Let me make some rays out. Okay. Okay, so this you can see pores like thing from where the water used to get inside it. So this pore cavity is your what? Is your ostia, from where the water used to get inside. Ostia, from where the water used to get inside, water in. Is it clear? Let me take another pen. Take the blue one. And the water got inside the body, then used to how used to get out? The water used to get out through this cavity, that is this portion it used to get out, that is called osculum, where water used to go out. Water out. Is it clear? And the cavity, this central cavity is known as your spongocoil. Spongocoil. Is it clear? And this spongocoil is lined by this cells. Okay, this cells will have the cavity that is ostia. It is lined by a cell called what? If it is disturbing, wait. It is lined by a cell called what? This spongocoil is lined by a cell called your collar cell. Collar cell, or you can say choanocytes. Is it clear? Are you understanding how the water used to get inside the body of a sponge through the ostia, which are pore-like thing present, okay, and how the water used to go out through osculum, okay, and the central cavity which is present, that is called your spongocoil, okay, and this spongocoil is lined by a cell that is called your collar cell or choanocytes. Is it clear? Sorry for the handwriting. Okay, is it clear to you all? Then we move to the next slide. So it is talking about they are having intracellular digestion, means inside the cell there is a digestion taking place. Is it clear? Then they are having, like us, we are having a skeletal structure, that is bones, cartilage we are having. So they are having a body skeleton that is made up of spongin fibers or spicules. This skeleton is made up of spongin fibers or spicules, we used to call it. Is it clear? Then coming to the next point, that is sponges are hermaphrodites, means sexes are not separated, means in one particular body both the male and female used to be there, means they are hermaphrodites, means sexes are not separate. Okay, then the next point we'll see that the reproduction mode of reproduction, the asexual mode of reproduction is through fragmentation, and sexual reproduction through the formation of gametes. Is it clear? Asexual mode of reproduction through fragmentation, and sexual reproduction in the by the formation of your gametes. Clear? Then moving on to the next slide. The fertilization it is talking about. Fertilization is internal. So internal, there is a fusion of sperm and ova. Okay, internal fertilization used to take place, and development of zygote does through a distinct larval stage, means the development is indirect, we can say indirect development. Indirect development, there is no direct development because they will have some larval stage in between, so development is indirect. Is it clear? So characteristic features is completed. Now let us see some examples. So this question has come many a times in your entrance examination, that is, which sponge is known as your freshwater sponge? This spongilla is known as your freshwater sponge. Spongilla, just remember. Okay, spongilla. Euspongia is your bath sponge. Euspongia is your bath sponge. Then you have cycons. Okay, just remember, spongilla is your freshwater sponge and euspongia is your bath sponge. Is it clear? And you have cycons also. The three examples you have to remember, these are important. Okay. Okay, let's see a picture of a spongilla. It is a freshwater sponge. Okay, it is a freshwater sponge. Okay, so we have completed the phylum number 1, that is Porifera. So in the next video I will bring the next phylum. All right, so thank you for your patient hearing.